Welcome to the second lecture on antimicrobial chemotherapy. Whenever we talk about the mode of action of antimicrobial agents, we need to look into the target sites of antimicrobial agents. The targets are to be chosen in such a way that these target sites are either absent onto the host cells or the target sites are altered in the host cells so that the drugs can execute their selective toxicity. In today's lecture, we will be specifically focusing bacterial cell wall biosynthesis inhibitors, cell membrane disruptors, and nucleic acid biosynthesis inhibitors. So let's start with bacterial cell wall biosynthesis inhibitor antibiotics. These drugs are supposed to be the drug of choice. The reason behind that, they show maximum chemotherapeutic index or maximum selective toxicity. The reason is quite obvious, that they are targeting bacterial cell wall biosynthesis. These cell walls are actually absent in eukaryotic cells, or our host system don't have cell walls. That's why they show maximum chemotherapeutic index. Now, if we try to precisely focus on bacterial cell walls, it comprises of polymer called Peptidoglycan. This Peptidoglycan is a polymer of carbohydrate and peptides.
Ma'am, how is this peptidoglycan synthesized within the bacterial cell?
Yeah, this is a very long process, but if we just go in brief, that it takes place in 3 different stages. Stage 1 takes place in the cytoplasm, wherein actually the polymerization of the carbohydrate moieties takes place. The polymer of carbohydrates are made up of N-acetyl-glucosamine and N-acetyl-muramic acid. And this N-acetyl-muramic acid actually is then attached further with 4 amino acids. This joining part is considered as the stage 1 of biosynthesis of peptidoglycan. Next stage actually takes place within the cytoplasmic membrane, wherein actually certain membrane bound enzymes are there. What they do, these membrane bound enzymes in this stage, actually there is exchange of this polymer of carbohydrates from UDP onto the lipid carriers. So these lipid carriers are lying on the inner surface of the cytoplasmic membrane. What they do actually, these lipid carriers actually help in carrying on this carbohydrate moieties through this lipid bilayers. Finally, the third stage takes place in the periplasmic space, wherein this uncross-linked or soluble polymer of carbohydrate and peptide gets linked together to a cross-linked polymer of carbohydrates and pentapeptides. Thereby, they make an entire meshwork of peptidoglycan. So this polymerization between the monolayers takes place in the periplasmic space. So in all, we will see that this process of peptidoglycan synthesis, there is involvement of at least 7 or more enzymes are involved. So these enzymes are actually chosen as the target sites by the chemotherapeutic agents. Most of the chemotherapeutic agents that inhibit bacterial cell wall biosynthesis actually target one or other enzyme which are involved in peptidoglycan synthesis. So the first key antibiotic that comes in this category is penicillins. As we all know that the entire credit of discovering penicillin goes to Alexander Fleming and Ernst Chain and Florey. So these penicillins are actually targeting the transpeptidation reaction, that is very much essential step in peptidoglycan biosynthesis. These penicillins actually contain important moiety called 6-amino-penicillinoic acid. So various derivatives of penicillins are derived just by changing the side chains attached to these amino groups. right? So the basic or the first penicillin that was discovered by Alexander Fleming was isolated from organism called Penicillium notatum. So this penicillin has got a very important ring that we call as the beta-lactam ring, is supposed to be the essential component that should be present in any cell wall biosynthesis inhibitors of this class. This cell wall biosynthesis inhibitors can be classified into two different categories depending on those one which contain beta-lactam rings and the other class which is non-beta-lactam ring containing or some other antibiotic targeted towards cell wall biosynthesis inhibition. So beta-lactam ring containing antibiotics actually are very much effective because they resemble D-alanine residues. These D-alanine residues are actually the last two amino acids which are attached onto the pentapeptide chain which is linked to the muraminic acid of the peptidoglycan. So what happens, actually they are just resembling D-alanine. This is the reason that the enzymes which are involved in linking this D-alanine onto the amino acid chains of muraminic acid, they instead of binding to D-alanine will bind to this beta-lactam antibiotics. And hence the attachment of D-alanine onto the tripeptide attached to muraminic acid is restricted.
Ma'am, you said that penicillins have this beta-lactam ring by which they carry out their activity. Is this the only method by which they can combat the infection?
No. They do have some other methodologies that sometimes they bind to certain periplasmic proteins called penicillin binding proteins. What they do when they bind to this penicillin binding proteins, the penicillin binding proteins are the proteins of the bacteria itself. They get modified, and what they do that they start creating holes in the bacterial cell. Thereby the cellular integrity is lost. This is another way whereby the penicillins execute their killing action. Naturally occurring penicillins comprises of two types. One is called penicillin G and other is called penicillin V. Actually penicillins were initially synthesized or they were supposed to execute their killing effect only on gram-positive bacteria. So the naturally occurring penicillins, obviously G and V, both are effective against only gram-positive bacteria. But the thing is, the basic difference between G and V is that penicillin G is destroyed by the acidic pH of the stomach. That's why you can't take penicillin G orally. You have to use it by injecting it intravenously or intramuscularly. Whereas this penicillin V is quite stable in the acidic pH of the stomach, so it can be taken orally. People have modified naturally occurring penicillins. They are called as semisynthetic penicillins. It means we have done chemical modification with the naturally occurring penicillins. It mainly comprises of ampicillin and amoxicillin. They are effective against both gram-positive ones and gram-negative ones. Here the examples are ticarcillin and piperacillin. But the good part about this extended spectrum antibiotics is that they are much more active against gram-negative bacteria. So they were targeted specifically against the Pseudomonas species. The first drawback of using penicillin as a drug is development of allergies. Many people are allergic towards penicillins. Some of them develop rashes on their skin, swelling of eyes, and many more side effects are associated with the use of penicillin. And the second problem with using penicillins is the development of penicillin resistance by the bacteria. What the bacteria actually does, as we said in our earlier part, that penicillins are active because of the presence of beta-lactam rings. So the bacteria start synthesizing enzymes that cleave these beta-lactam rings. So the gene that codes for an enzyme called penicillinase. So what it will do? It will cleave the beta-lactam ring, thereby it will inactivate the penicillin. and the bacteria will start showing resistance against these penicillins. We have synthesized certain drugs like methicillin or dicloxycillin. They have got such beta-lactam rings which cannot be cleaved by this penicillinase produced by the bacteria. So these drugs were initially launched to overcome or to combat this staphylococcus which was capable of producing penicillinase. Right? Later on we could find that this bacteria have started developing resistance in a different way. When they could find that the penicillinase that they are producing is unable to take care of the modified beta-lactam rings, then what they did, they started synthesizing certain altered penicillin binding proteins. As I said, that penicillin binding proteins are the target sites of the penicillins. So this resistant bacteria have started synthesizing a modified penicillin binding protein which not at all binds to penicillin. So the tug of war is always going on, right? We people are trying to modify the drugs to combat the pathogen, and pathogen is also modifying itself day by day to combat the toxic effect of the drug. Now we go for a combination of two drugs. This type of drugs are called penicillins plus beta-lactamase inhibitors are clubbed together and are administered together. So such a drug is called augmentin, wherein what we have got amoxicillin and clavulanic acid combined together. What clavulanic acid does? Clavulanic acid will take care of the beta-lactamase enzyme produced by the bacteria, and then amoxicillin has got its beta-lactam ring intact. Then amoxicillin can execute its cell wall biosynthesis inhibition action very nicely. People who are allergic to penicillins, we do use some other beta-lactam antibiotics such as cephalosporins, monobactams, carbapenems. So let's start with cephalosporins. Cephalosporins are similar to penicillins, that they do have beta-lactam ring and they also target the transpeptidation reaction. But they are better because we do have developed a class of cephalosporins, say first generation, second generation, third generation and fourth generation, wherein we will see that initially the first generation cephalosporins were specifically more active towards the gram-positive ones. Then we have developed the second generation cephalosporins which are active against gram-negative ones, and anaerobic bacteria were also killed by second generations. Then we developed third generation wherein we could see that they were active against gram-positive, gram-negative, as well as they could cross the CNS, means blood-brain barrier they can cross, and they can be used for the treatment of meningitis or like this. Then we have got the fourth generation antibiotics which had a broader range of activity, and what benefit we got here, that they were active against the opportunistic pathogens. That was an additional benefit that we got by using cephalosporins. Monobactams are also beta-lactam antibiotics, but the basic difference is that it contains only one ring, one beta-lactam ring. Unlike the penicillins where we do have got two rings, in monobactams we have got one ring. It has got, the drugs are called moxlactam or azditonum. So this drugs were specifically targeted towards the gram-negative rods actually. This is the benefit that if we are very much knowing that what is the causative organism, then we will be specifically using moxlactam or monobactams. And if the diseases are not specifically known or the causative organisms are not known, then we prefer to go for broader spectrums like cephalosporins. Third group of beta-lactam antibiotics is called carbapenems. They are comparatively broader spectrum antibiotics, and what is the benefit of carbapenems, that they can be used for patients who are allergic towards this penicillins. But there was a drawback with carbapenems, that whenever this type of drugs are taken, they are broken down within the kidneys. So this is the drawback of using carbapenems. this drug, that is impenem, a derivative of carbapenem, is mixed with another drug called cilastatin. So what is done, this cilastatin and impenem, they together are called as Primexin. This drug, where cilastatin will prevent the cleavage or the breakage of the compound within the kidney cells, then impenem can execute its killing action. Now let's focus on some glycopeptides that inhibit cell wall biosynthesis. To start with, vancomycin. It is a cup-shaped molecule wherein a peptide is linked to a disaccharide. These antibiotics are effective or bactericidal against streptococcal, staphylococcal. They are also blocking the transpeptidation reaction of the cell wall biosynthesis, utilizing the peptide backbone of the antibiotic.
Ma'am, we have heard that vancomycin is called as the drug of last resort.
Yeah.
Why is it so?
Because it is active against those bacteria, Staphylococcus and Enterococcus, that are showing resistance towards many class of antibiotics. So when other antibiotics are becoming non-functional or bacteria starts showing resistance against them, then we go for using vancomycin. Teicoplanin. It is also used for as a cell wall biosynthesis inhibitor. It is active against Staphylococcus, Enterococcus, Clostridia and many gram-positive pathogens. Its mode of action is similar to vancomycin, but it has got fewer side effects. Its side effects are a bit less. Bacitracin is a narrow spectrum antibiotic which is produced by Bacillus. It actually prevents the elongation of the peptidoglycans in gram-positive bacteria.
Ma'am, how bacitracin blocks the elongation of the peptidoglycan?
Actually, it prevents the transport of the peptidoglycan precursors across the cytoplasmic membrane. This is how it executes the inhibition of cell wall biosynthesis. Cycloserine is also a broad spectrum antibiotic. It can be bactericidal as well as bacteriostatic depending on the concentration of the antibiotic and the site of infection. It may be bactericidal sometimes and sometimes it is bacteriostatic. It also inhibits two different enzymes. One is called L-alanine racemase, which is responsible for synthesis of D-alanine from L-alanine, and the second enzymatic pathway that is being inhibited by cycloserine is called D-alanyl alanine synthetase. Both the enzymes are being inhibited by cycloserine because cycloserine is actually an analog of D-alanine. That's why it inhibits these two pathways. Phosphomycin, it is also a broad spectrum antibiotic that is targeted and that is also a cell wall biosynthesis inhibitor. With this actually we complete the journey of bacterial cell wall biosynthesis inhibitors. Now we will be discussing about bacterial cell membrane disruptors. The most important drug that is used for as cell membrane disruptors are Polymyxin B, and they are basically cyclic glycopeptides that insert themselves into the cell membrane. They actually scavenge the magnesium ions and the calcium ions that were present with the phosphate groups of the cell membranes. Thereby, they are acting as a detergents and they liquefy the cell membranes, wherein the integrity of the cell is lost and the cell dies. So, this type of drugs are very much essential in bacterial killings, but we do have got certain drawbacks that they may cause kidney damages and sometimes they cause breath arrests. In this category, it can be divided into two groups. First group will be the antibiotics that prevent the DNA replication process. Other group will be the antibiotics that prevent the transcription process, means conversion of DNA to RNA. So let's start with the transcription inhibitors. DNA transcription inhibitor comprises of first antibiotic called Rifampicin. Rifampicin executes its action by binding to the beta subunit of RNA polymerase. RNA polymerase is the enzyme that carries on the transcription process. So what rifampicin does, that it binds to the beta subunit and blocks the transcription process. It is a bactericidal antibiotic which is having a broad spectrum of activity. It is used specifically for the treatment of tuberculosis and for eliminating meningococci. Rifampicin has got certain limitations, that it can cause certain kidney damages or liver damages, and it has been seen that if a patient is being treated with certain anticoagulating agents are being given, then the dosage of anticoagulating agents are to be increased when rifampicin treatment is going on. It is a drug that prevents the transcription process by binding to the DNA. It specifically binds to the guanosine and cytosine linked DNA, means GC residues are the sites wherein actinomycin D will execute its action. So it binds, or it gets inserted into the major groove of the DNA, thereby it prevents the transcription process. So what we have seen, that we do have got two transcription inhibitors. One is called rifampicin, which binds to the beta subunit, and we do have got another antibiotic called actinomycin D that binds to the double-stranded DNA, thereby preventing the transcription process. Now we will be discussing about antibiotics that prevent the DNA replication process. Quinolones are actually synthetic analogues of nalidixic acid. They actually prevent the process of replication by binding to enzymes called DNA gyrase or topoisomerase 4. The function of DNA gyrase or topoisomerase 4 is to catalyze the unwinding of the DNA, which is an essential prerequisite for execution of DNA replication process. So we can see that they are supposed to be broad spectrum antibiotics which are effective against gram-positive bacteria as well as gram-negative bacteria. The drugs, they contain a 4-quinolone ring. The drugs which are usually used are norfloxacin, ciprofloxacin, levofloxacin, and these type of drugs are specifically targeted for the treatment of traveler's diarrhea, urinary tract infections, and wherein many resistant bacteria can be combated utilizing quinolones. They are also effective in against many sexually transmitted diseases, many respiratory disorders. They are usually used. So if we look into the drawbacks of quinolones, it's the two important drawbacks are excessive use of quinolones actually leads to development of resistance against quinolones, number one, and many a times people taking too much of quinolones develop certain brain disturbances and seizures. We dealt with bacterial cell wall biosynthesis inhibitors. We focused on beta-lactam antibiotics, then we focused on antibiotics which are non-beta-lactam ring containing or glycopeptides. They also inhibited cell wall biosynthesis. Then we discussed about membrane disruptors. Then we focused about nucleic acid biosynthesis inhibitors. In this category, we have taken up the transcription inhibitors as well as replication inhibitors.